Okay, so let's talk about stem cells. So what makes a cell a stem cell? There's really two key factors. The first is that a stem cell can continuously replicate itself. So it's able to continuously generate new cells. And that's different from a differentiated cell like a liver cell or a skin cell that has a limited number of times it can continue to divide. There's something called a Hayflick limit, and it's thought that a differentiated cell, assuming it is one that replicates often, can replicate about 50 times. And then what happens is, if this is your chromosome, each time your cell replicates, you lose a little bit of DNA on the end. And so each time your chromosomes are replicating, they get shorter and shorter. And it reaches a limit where the ends of the chromosome are getting too close to the gene coding information. And so the cell will actually undergo apoptosis, which is programmed cell death. And so there is a limit for how many times a differentiated cell can divide. Okay. In stem cells, the enzyme telomerase, which is the enzyme that makes the ends of the chromosome, this enzyme is functional in stem cells. So even though they're replicating, they have an enzyme that will keep their chromosomes the proper length, so there's no shortening. In a differentiated cell, the enzyme telomerase is off, and that's why the chromosomes get shorter and shorter every time the cell replicates. That's why differentiated cells have this Hayflick limit. So one of the key components of a stem cell is that it can replicate itself forever because it has that enzyme telomerase activated. The other key thing about a stem cell is that it can differentiate. Which means it can become different cell types, right? So it might become skin or muscle or nerves or blood or fat or immune system cells. Okay. And the amount of differentiation it can do, well, let's just talk about it on the next slide. So stem cells need to be able to replicate themselves forever, and they need to differentiate. So it's kind of like, I don't know what I want to be when I grow up, and okay, now I'm going to be, a teacher, scientist, an engineer, whatever. Once you choose that path, you can't go back. Now, that's not true in life, but that's true in cells. Okay, so that brings us to the different types of stem cells. So this type of stem cell with the most, what we call potential, is totipotent cells. Okay. That is here, 1 to 3 days of an embryo. Okay, very brand new cell, has not had any signals to start differentiating at all. We can't seem to capture these kinds of cells, but we can capture what we call pluripotent stem cells. Okay. So now you have cells that have differentiated just a little bit into the three types of basic cells. So you have the endoderm, the mesoderm, and the ectoderm. Okay. These are pluripotent because they still have lots of potential. Right? The endoderm line can differentiate into lots of different cell types. The mesoderm has its own cell types. Ectoderm, again, other cell types. And I tell you, I'm not a developmental biologist, so I honestly don't remember all of the options. But these are going to be our embryonic stem cells. Okay? So you can see here, you can get pluripotent stem cells at a week to 2 of a blastocyst. So as that little ball of cells is developing, you can get these cells that still have lots of potential, still can develop or differentiate, into many different cell types. Then they differentiate a little bit more, and you have what we call multipotent stem cells. Okay? And these are also called adult stem cells. So they are kind of have a destination. So for instance, bone marrow is a multipotent stem cell. It can still differentiate into lots of different types of immune cells and red blood cells, white blood cells, but it couldn't make lung cells. It could not make skin cells. So its choices are still a little limited. Again, you are full of adult multipotent stem cells. The only problem is, though it's fairly easy to get bone marrow, I couldn't make skin or nerves, or in this case, lungs or pancreas from that type of cell line. I couldn't even make muscle cells. So pluripotent, as far as the potential to differentiate into different types of cells, has the most potential, and that's what people are fairly interested in. And you will see, we will talk about induced pluripotent stem cells, which is basically taking an adult cell and making it kind of revert back. Okay, so again, this is just a nice overview. So types of stem cells and where they come from. Embryonic is coming from the blastocyst of a recently dividing embryo. Tissue-specific stem cells come from adults, and adult really just means once you're born. And induced pluripotent stem cells we'll talk about is where we take an adult cell and we revert it back to a pluripotent stem cell. Pretty cool stuff. So let's look at each of these one by one. Embryonic stem cells are, this is the inner cell mass. These cells are harvested, and remember, these are pluripotent. Okay, so they've got lots of potential of what kinds of cells they can come from. And these are from in vitro fertilization, what we're going to call leftovers. Okay, so if people are going through in vitro fertilization, a lot of times they will make a lot of, maybe 10 or 20 embryos. And then they implant a few of those embryos to try to make a pregnancy happen. So there are lots of these little blastocysts left over from in vitro fertilization that people can then donate to science, and we have these embryonic stem cells. Okay. There's been a lot of regulations on this because, yes, in theory, this blastocyst could derive a human being. Okay. Nobody is taking these and trying to actually grow a human in a petri dish, but it has the potential, and that's where the ethical issues come up. We can talk about, that in class, but it's 1 to 2 weeks of cell division. The blastocyst is about 150 to 200 cell stage, and you can get these pluripotent stem cells, grow them up in the lab. So there are commercially available stem cell lines that have been created before the government put a moratorium on government-funded embryonic stem cell research. Though these cells have the most potential, you have to remember that if we're going to use them for human therapy, they are not your genetic makeup. So this is like getting a transplant organ in that your body might reject it. That's why the idea of induced pluripotent stem cells is so exciting. Okay, so that's embryonic. Where do you find adult stem cells? Lots of locations in the body. You do have skin cells that are stem cells. That's why you can constantly shed your skin and create new skin. There are some in the heart that people are harvesting to try to repair heart muscle. Bone marrow is probably the most common one. And different places, there's even some in your lungs. I love the teeth because I used to tell my kids that I could clone them when they lose their baby teeth, and it freaked them out. The hard thing about adult stem cells is they're very low in number. They're hard to isolate. So they're hard to get access to. Bone marrow is the easiest one because you can just stick a big needle into your femur, into your big leg bone, and suck out some of that bone marrow. Hard to isolate, get access to. They're difficult to grow in culture or in the lab. I'm just going to unplug this because the light keeps going on and off. Okay. And these are multipotent. So they can differentiate into some types of cells, but not all types of cells. Okay. So we have embryonic, we have adult, and I keep emphasizing that pluripotent are so important because they have the ability to diversify. So once you start becoming even an adult stem cell, you start getting epigenetic markers that turn off some genes, turn on other genes, and that's really what's limiting your potential. And so it's hard to reprogram these adult stem cells where pluripotent don't have these epigenetic marks, and so they are ready to get signals from the environment, become this kind of cell, become this kind of cell, become this kind of cell. So like I said, kind of the new embryonic stem cells is this idea of induced pluripotent stem cells. So we want pluripotent stem cells because they can become all different types of cells. Normally we have to get them from an embryo, but we are now learning that we can induce them back to a pluripotent stage. So how does that work? So induced pluripotent stem cells, what you have to do, and the research for this started in 2006. And what they found was that they could add several genes. Okay? And the ones that are the most used, KFL4, SOX2, CMYC, and OCT3-4. And then depending on whose paper you read, Nanog, Lin, there's a MBD3. Different labs use different genes that can take an adult cell. So a fibroblast cell is a cell that helps make connective tissue in your body. They can reprogram it and make these pluripotent stem cells that then can become any type of cell we need to regenerate in the body. What most of these genes are transcription factors. Oops. see if this will work. Okay. So transcription factors, remember, turn on genes, control genes. And so adding these genes kind of help turn back on genes that were silenced as cells differentiated. The challenge that many, many researchers found is that sometimes when you turn and reprogram back these cells, they actually become cancer, which cancer in this case means they're dividing uncontrollably. So you don't want to put those kinds of cells back into a person. The other challenge is how you're going to deliver these genes efficiently to these cells. So I'm going to show you this. And this is a cool video down here talking about the guy who kind of first was able to make these induced pluripotent stem cells. So I encourage you to watch that when you get a chance. Here's a little bit about the history. So to reprogram these cells, we have to deliver genes. And we've talked about viruses are really great at delivering genes. We've talked about this for gene therapy. The problem is, we also talked about this in gene therapy, you can't control where the DNA is integrated. That's another reason you can get cancer by reprogramming these or making these induced pluripotent stem cells, because you might turn on an oncogene or turn off a tumor suppressor gene or kind of mess something up in the cell cycle. So they've done lots of different, so Krelok's transposons are another way to integrate the DNA. The second generation was non-integrated DNA, and what I want you to see is the efficiency went way down. Okay, so they were all, you're only talking like 1% efficiency, and now you're down to 0.001% efficiency. So this is really not good. This means it's taking a lot of resources and a lot of time to try to reprogram these adult cells to pluripotent stem cells. One kind of looks kind of good technology that Sigma Aldrich is working on is actually using RNA as a reprogramming. So what they're doing is they have this RNA molecule. Polycistronic means many genes in one RNA. And you can just transfect. So remember when we talked about transformation of putting plasmid DNA into bacteria? Transfection's the same type of thing, but for eukaryotic cells. So now they're putting RNA into this adult cell. It's turning on all the genes it needs to, so it's getting translated into protein. The proteins are turning on the genes, and then you can actually, you remove some kind of drug, and the RNA will be degraded, and now you have induced pluripotent stem cells that don't have any of these transgenes in them. So it's pretty cool. Possibility. I have not had a chance to look into the research how far this has gone. But remember, the excitement over induced pluripotent stem cells is that if you need cells to repair, you want them to have the same DNA as you, so that you have a lower chance of rejecting those new cells. Okay, so we're trying to revert them to embryonic stem cells, but with your same DNA. There's one other way to make embryonic stem cells, and it's called somatic cell nuclear transfer. Okay. And so this is where you take one of your cells with your DNA, say a skin cell, and you take the nucleus and you put it into an egg that's been enucleated, so they've removed the nucleus of the egg, right? And then you make the egg divide, because this is already diploid, so it doesn't need fertilization because you are diploid. And you can generate embryonic stem cells. Okay. You need egg donors for this. And from what I've heard, it is not easy to donate eggs, and most people that donate eggs are doing them to help couples conceive. But this again is another way, and honestly I forgot to even look up what the efficiency is. I'll try to find that before class. Making myself a note. Diploid. What was I saying? Anyways, these stem cells have your genetic information. They do have mitochondria from someone else, so you do have genetic information from the egg donor. So in theory, yes, you could donate your own eggs. Most of the time when we're to that point where we might need stem cell therapy, we might be a little old. We might not have producing eggs, and then obviously that wouldn't work for guys. You guys don't produce eggs. But this is another way to make embryonic stem cells. I believe iPSCs are kind of the more hot topic for research. Okay, so super, we can get these cells. Why? Why do we want to make them? And what we're talking about is what we call therapeutic cloning. So we are not doing reproductive cloning. But when we come back after break, we will actually talk about somatic cell nuclear transfer as a way to clone animals. So Dolly the sheep was made this way. But we're not doing that with humans, at least hopefully not. But what we are doing, again, is somehow taking your adult cells, making them into embryonic stem cells, whether it's through SCNT or iPSC, so that we can regenerate tissue for you. And that's what therapeutic cloning is. So maybe you need some new muscle cells or some heart cells. You guys are reading about different stem cell research and how they're using it to treat things like diabetes and macular degeneration. So we'll talk more about the applications of therapeutic cloning. We'll discuss that in class. But that's one of the reasons stem cells are so exciting. You are also going to look up stem cell, well, this is for stem cell clinical trials. So this is a link, the clinical trials, you've looked at that website before. This is a link to specific stem cell trials, sorry. But you're also going to look up some of the commercial applications of stem cells. Okay, so there are places around Denver that you can go and get stem cell therapy. You don't need a prescription. This is this the one? Oh yeah, next slide. So these were some headlines I had found. This was pretty cool. Regenerating a cornea to make a healthy eye for someone. 6 years after treatment, they were able to see again. They talk about repairing heart and repairing cartilage, diabetes. You're going to read about some of these. There's a cool TED Talk about this regenerating cardiac tissue with stem cells. But there's also other commercial ones that might not be as scientific. So you can get a facelift. You can look young again. And what they're doing with this type is they're taking out your fat stem cells and they are putting them into places to help stimulate new cell growth. Okay, so a facelift, things for arthritis. This is one you might want to look up. This is with dubious stem cell clinics, which is why I put this like stem bucks thing. So with any kind of technology, there are applications that may or may not be very scientifically approved. So that's your other part of your research for the discussion. This is a place in Johnstown, Colorado that does a lot of stem cell. And so we're calling this regenerative medicine because you're trying to regenerate cells. Therapeutic cloning, regenerative medicine, they kind of both go together, right? So you can look up on this site about stem cell therapy that you can just walk in, pay your money. I don't know if it's covered by insurance, but see what kind of information you can find and share with the class. The last thing I just want to give you, you may run into these terms. So if you read anything that says allogeneic, means that you have a matched donor. So this is like when you're doing a bone marrow transplant, you get it from an allogenic donor. They're matching as best they can. An autologous, so auto means self. So automobile is self-moving. And so this is getting the cells from you. Okay. So this describes your source. These describe the use. So homologous, homo, same. So this means you're using the same type of stem cell as target treatment. So this again would be like taking bone marrow stem cells and using it to replace T cells or B cells or immune cells, right? Non-homologous is stem cells from a different tissue. So stem cells from one tissue being used in a different tissue. So this is like taking your fat stem cells and putting them in the joints of your knees to help with arthritis. So non-homologous, not the same. What I think is really cool is there's a lot of potential research looking at the use of stem cells. So things in the brain, right? Blindness, deafness, heart, muscular dystrophy, diabetes, cancer, spinal cord injury, bone marrow transplant, wound healing. I love missing teeth. I haven't really looked into that, but that'd be kind of fun to grow some teeth. So this is the fun stuff we're going to talk about in class, the potential uses that you have found and you have learned about for stem cells. So remember the summary or the goal of stem cell therapy is to fix tissue, whether it's degrading because of old age or overuse, or you lose something due to an accident. There are different ways to generate your stem cells. So you can do the reprogramming, the induced pluripotent stem cells. You can get embryonic stem cells. You can use adult stem cells, all depending on your application. Okay. And then, again, the whole idea is to make cells to help fix your body. So that's why we call it either therapeutic cloning. And the reason cloning is used is because this is when you are making the exact copy. So this is really with somatic cell nuclear transfer. Or we talk about regenerative medicine, where you are, oh, I hate this pen, creating new tissue to fix or repair. Okay, that's what I have for you. I apologize for this being so delayed this week, and I look forward to our discussion on Monday. Bye.